So this is one of those quotes that I always tried to wrap my mind around. And when I finally read the paper, to understand where this guy was coming from, it's an interesting paper. And he makes some claims that that we now understand maybe don't fit the evidence as well as they did at the time. But this has become a mantra of sorts. And the reason this came to my attention, this particular quote was used very heavily by an atheist philosopher named Alex Rosenberg. He's a philosopher of biology. And last year I was asked by Dr. Ralph Gilmore at Freed-Hardeman to help him prepare for a debate with Dr. Rosenberg. So as I read Dr. Rosenberg's writing, this is what he calls Dobzhansky's dictum. And this is what he hinged a lot of his ideology on. So I read the original paper and all those things up with it. I didn't realize that was actually the title of the paper. It didn't. I thought this was a quote in the paper, but this was really more the overall theme of the paper. But at any rate, this is very much the way we're trained nowadays, right? This is in grad school where I went to Vanderbilt. This is how we train students and what it looks like. And then this is another concept that permeates the training in biological sciences. So Francis Crick in his writing says this, hey, come on in. Welcome. Good to have y'all here. Francis Crick, co-discoverer of the structure of DNA, right? The double helical structure of DNA. Suggested biologists must constantly remind themselves that what they see was not designed, but rather evolved. And that's a really bold statement, right? But it says something about the viewpoint, the paradigm as is discussed in the film, that science is approached from. You have to look at it and say it was not designed. Now, if I asked you to walk in this classroom and, it looks like Starship Enterprise, like if you saw the controls back here, you think we could take off, but this is mission control. But if I asked you to walk in this classroom and told you, hey, by the way, nothing in there was designed, it's completely an accident, you would say, well, that's crazy. There's seats, there's tables, everything just seems to be in just the right place. And as a biochemist, when someone asks me to believe this, I feel the same way. I look at it and I say, I just have a real hard time doing that because things seem to be in the right place. The equipment all seems to be in place to do the job that it's tasked to do. And that to me is an amazing thing. I was working over the last few weeks, a couple months really, with some kind of distant family members and friends of mine who I went to undergraduate with at Freed-Hardeman who have ended up in the computer science realm. And my interest is in learning from computer scientists and engineers. Are there concepts that they use that we can learn from in the biological sciences realm? And one that came up that Dr. Tackett I'd like for you to come up and lecture for a few minutes on, is the concept of layering. So I came to learn about layering from our colleagues in the computer sciences realm. The idea being, one of the biggest challenges, in my understanding, in the design of computing information systems and software systems is that of complexity. And in order to simplify complexity, we use layers. Is that an appropriate way to state that, Dr. Tackett? And so layering allows us to separate things out. And these things happen all the time, right? Open your web browser and go to Google, right? Type in Google. And what happens is, all of these things, all in order, right? Back and forth as it sends information out and information comes back and your screen populates, and this is occurring in what? Milliseconds, right? Ridiculously fast, considering all of the steps that are taken. The only way that works, though, is if all those layers, one on top of the other, operate in their own space and do the right thing in their own space. And what's interesting about that, and I may have time to talk about this later, depending on how much time we have, there's a lot of analogies to that, to the genome in particular, and I'll talk about that later this week. The genome has a modularity, I believe, that is very similar in encoded structure to our modern information systems, where you might have a packet, an address header, etc., before the actual data. It's very similar to what we see in the genome. And if we can look at the genome from that perspective, as in instead of this non-coding information being there by accident, if it's there by design, the question is what's it doing? And how can we use that to learn about how the system works? So layers of organization occurs at the biological level, right? So we see this on many different levels, right? An organism is made of organ systems, at least humans. We have organ systems, and those organ systems are made up of organs. And so we've got organs, Organs are made up of tissues. Tissues are made of cells. Cells are made of subcellular organelles like the nucleus, which contains things like DNA, which is made of atoms. And that's not an official picture of an atom, but it works, right? It's cartoony. But you get the point, right? There are layers of organization in biological systems. And we could do this analogy in several different ways, but just to stop and think about it, an organism has all these lower layers. And that's how we deal with the complexity, right? How is it that we see and hear and feel and touch, move around, all the different things that we do? It has to do with the various layers, right? So we have a skeletal system, we have muscles, we have a respiratory system, we have a digestive system, etc. And each of these are organized in different layers. And so within our body, we've got however many different, I don't know how many systems there are and organs there are, but each of those are made up of sets of tissues and different types of cells. And depending upon who you talk to, we have a couple hundred different types of cells throughout our body, and maybe more than that, depending on how specialized you want to be. So several hundred different cell types that all came from that original egg and sperm that fused, right? So it's a pretty amazing thing when you think about all of that encoded information that comes out in this whole process. And to think about the compartments within the cell, the nucleus, and then the molecules within that, DNA and other proteins and RNA, all of that has to come together to make the system work the way it does, which is really amazing that any of it works as well as it does. So, life at its basic level is cellular, right? We all understand this. On a bottom line level, life is composed of cells. Right? You could argue that there are some smaller organisms maybe, but generally speaking, this is considered the basic unit of life in most textbooks, and I think in practice that's, I think, still fair to say. Yes, go ahead.
What is often meant by life?
What is meant by life? Now you've gone into philosophy. I'll turn you over to Dr. Wise.
The reason why...
Yes, that's a good question.
It seems like this is, from articles that I've read from evolutionists, they hinge upon a certain definition of what life is to argue that we don't have life today. It's just, again, it's a perception. Like we have a perception of design. Life is a perception.
Right.
Is a cell living? It's those.
Right.
So
right. No, and that's a fair question, because I think there's a philosophical definition, right? But there's also can be operational definitions. But it's the classic thing. You walk into the first day of biology class and the teacher asks, "What's living?" Right, Dr. Fader? What's living? Right? And what are the characteristics of a living system? And in my realm, I think of humans and human cells, and I think there's a pretty clear definition from that perspective of what we would consider living. Where it gets murkier is as you get to the more fundamental forms of life and things like that. Now, the question you're asking about is almost a little bit different, and that is, are we actually alive in the sense of consciousness and knowing what's going on? And to me, that's a separate question from whether or not a cell goes through its biochemical processes. Does that make sense?
So an organism going through its biochemical operations would be considered scientifically living.
Yeah, I think we could, generally speaking, consider a living organism is something that, it's in operation. The cells are not at the state of highest entropy, so to speak. In other words, we've got replication, transcription, translation. We have digestion of food particles to make energy. All of these different components allow that cell to be alive, as we say it. Now, on a separate level, what you're saying and asking about, I think, is more the idea of consciousness and living, and how is it that we can stand here and say, I know that I'm here, and I know that you all are there, and I know I'm alive, and I know you're alive. That's a very different question from the biochemical question of living. If I think that's a fair way to say that. But that's what I came up against when I was reading a lot of Dr. Rosenberg's writing, preparing for the debate that Dr. Gilmore had with him. Was Dr. Rosenberg believes that all life is physical. So there is nothing outside of the physical realm. There's nothing but physical. And so therefore, all life is essentially boiled down to chemistry and physics. And if that's the case, then you and I can never truly be alive. Our cells are just operating out the laws of chemistry and physics, and that's it. And so you aren't really sitting there thinking about me talking. You just are under the illusion that you're sitting there thinking about me talking. I think that's probably about the best way to say it.
Right?
That's right. Yeah. If you're even here. Yeah. If you even exist, right?
Okay.
It gets you way out. It takes you way out there. But no, that's a fair question because it is an important distinction. When we talk about a cell being alive, right? We could go into the laboratory. We could go to Dr. Stevens' laboratory at Freed-Hardeman and look at cell cultures and determine, is this cell dead or is it alive, right? It's going to have certain characteristics. But that's a separate question from the atheist who's maybe a physicalist that believes that all that exists is physical. They're going to say, well, that cell may be undergoing these things, but it's not really, you're not really alive observing it because you're just playing out chemistry and physics. So no, that's a good question. So I'm not going to go through all the organelles, but you understand it's cellular and then even subcellular and compartmentalized. And each of those subcellular compartments has fundamental biomolecules, right? So you've got amino acids, which make up proteins. You've got nucleic acids, which are our DNA and our RNA that we talk so much about. But you also have two other classes that are really important. You've got lipids and carbohydrates. And so, where do we usually interface with this? It's usually on the side of a cardboard box that we bought to eat, right? So it's our cereal box, it's our box of mac and cheese, whatever, right? It's funny, if you look at the list next time, you'll see protein, lipids, and carbohydrates. You won't see DNA. For some reason, we don't have a daily requirement of DNA, but we'll get to that later. But yeah, we eat a lot of it, but it's not required in our diet, and I'll show you why that is in a few minutes. But lipids are really, really critical, right? The fatty acids, they could be, steroid-based molecules like hormones. These are really critical. So cholesterol is actually really, really important. We've made it out to be the bad guy, and there's some problems that come along with how it gets processed in our body maybe and how it gets packaged. But ultimately, we have to have cholesterol. If you don't have cholesterol, you die. And so you've got to have it. Right? And so fat in and of itself is not evil, but there was a war on fat, right? Decades long war on fat, fat-free everything. What do we do when we remove fat from everything? Put sugar in it. So then there was a decades long carbohydrate kick, and now we're on the war on carbohydrates, right? So now Atkins and all these other diets have come out. You need to get rid of carbohydrates. They're evil. They're terrible. I think if we step back out of it, God designed this world in a way that we can consume the food that's out there. Problem is we keep modifying it, putting way too much fat in it or way too much carbohydrate. Or we just can't seem to find that moderation balance. And we end up with these diets that are really high in this or really high in that or really low in this, where I think we can be more balanced about it. I say that just to say those fundamental components are actually what are used to build all of the cells in your body. You've got to have fats in your diet. You've got to have proteins in your diet. You've got to have carbohydrates in your diet. You need to. So it's not an unhealthy thing to do. But there has to be balance to that. And our body has the ability to make everything. Now, one of the things that I want to point out here is that all of these biomolecules are fundamentally interconnected. And this is a really interesting phenomenon. So lipids and carbohydrates can actually be synthesized and metabolized by proteins in our body. Now, we can't build every type of lipid that we need. There's some that we have to get from our diets, but most of them we can make. Our body has the ability to produce. Carbohydrates, we can actually make sugar if we need to. Now, it's not preferable. Your brain's going to get really angry if you don't have any carbohydrates coming in, but we can do it. But so where do we get proteins? Well, proteins are made by proteins and RNA. Where do we get RNA? Well, we make that from DNA using proteins. All right, where do we get DNA? Well, we have nucleotides, which are made from carbohydrates and amino acids synthesized by protein enzymes. So you start to see there's a problem here with the system if you're trying to build it from little bitty pieces together. In other words, God designed the system to all operate together. And so here's the metabolic map. And this is Stanford University's Pathways map version. You can download this for free online. I show this not to be overwhelming, but just to help us recognize there's an interconversion of molecules throughout our cells that goes on a daily basis without you thinking about it, without you telling it what to do. It is dynamically adapting and responding to what food you have, how much you eat, how much activity, All of these things are ongoing, dynamically regulated. And so we've got, for example, the carbohydrate pathway is kind of through the middle here, which feeds into an energy production pathway, which is where we make all of our ATP right down there. But let's say we don't need energy right now. Let's say we need to make amino acids. Well, we've got a whole area over here on which amino acids we can make, how we can make them. Or let's say we need to make nucleotides. We've got a whole section over here on nucleotides. What about lipids? We've got lipid synthesis over here. So we have all these different pathways, and ultimately they're interconnected. There's feedbacks, there's connections, there's cross-links. And so on a fundamental level, and I'm not just saying this as a biochemist, I love biochemistry, but on a fundamental level, life is dependent upon this interplay of biomolecules. It's really, really critical. And so there's a lot more to it, but yet it's a really elegant system. In fact, I would love to sit down with this and some engineers and computer scientists, because I think there's a way to map this out differently to help us understand more about what's going on in the interconnections between these pathways. Because what you'll see in different places here is that there are ways to go back and forth between the carbohydrates and the lipids. There are places to go back and forth between the amino acids and the carbohydrates. There are places of interplay even with the nucleotides, and they're strained to show it on here. I was trying to find the connection. They don't show it on this map very well, but actually amino acids feed over into the nucleotide pathway to build our nucleotides. So we've got this entire interplay. All of these things are interconnected. So while I talk about them as discrete molecules, they are, but our body can interchange them back and forth depending upon what we need. So if I'm not getting enough of a certain thing in, my body can say, we don't have enough of that. Let's use some of this and make that. And for the most part, we can do that. Now, proteins, there's an area where we can't, right? Because certain amino acids we cannot synthesize, so we're dependent on our diet. So this interdependence of biological molecules, this is what a friend of mine calls causal circularity. Biological molecules are inherently interdependent, and we usually depict this using the central dogma, and that's this idea that DNA in our cells is used to make RNA, which is used to make protein. So all the DNA in an organism we call the genome, all the RNA it makes we might call the transcriptome, all the proteins that it's going to make we might call the proteome. So DNA gets transcribed, so we call it transcription to make RNA. RNA gets translated to make protein, so we call that translation. If we're copying DNA, we call that replication. So we've got these fundamental processes. Well, it turns out replication requires numerous proteins in RNA. Transcription requires proteins in RNA. Translation requires proteins in RNA. So any one of these steps, and I'll show you a table here in a few minutes to depict this a little bit further, but any one of these steps is dependent upon the other components to fulfill the pathway. So the products of the processes are required to complete the processes, which is a hallmark of an engineered system, Dr. Tackett. It's the concept that all of these components have to be in here together for the system to operate, which is important when we're talking about origins, because it begs the question of how did the first system arise? And that's a real big challenge for our evolutionary colleagues to try to wrap their mind around how you get to this state where now all of these components are so heavily dependent upon one another. And people like Rosenberg and others have tried to envision a self-autocatalytic molecule that just brings itself into existence and makes more of itself, etc. And that's a nice thought. But that assumes that you're in a laboratory in a very controlled environment with a lot of substrate material available and nothing to disrupt it, perfect conditions. There's a whole lot that goes into that would be required for that system to ever be possible. So I think that the system it just was not designed that way. Just really quickly, DNA. So you're familiar with DNA as a double helical structure of 4 bases, adenine, thymine, guanine, and cytosine, which base pair together as A and T and G and C. And they base pair across the double helix. So we have, this is kind of looking from the end on the spiral of it, but if you think about it, there's base pairing going on between the 2 strands. And you can actually separate those strands, and it's a really interesting process. That's something I've studied in my research. But the bases are arranged in a specific order. We call that a sequence. And it's that sequence that is the information that we're talking about that tells the cell, here's how to build this protein we need. Here's how to make this structure. An interesting question that I think will come up and has already come up, but will continue to come up, is how much of the information in biological systems is encoded in the DNA and how much is outside the DNA. So I'm of a mind to believe that not everything that's needed to build a cell is actually encoded in the DNA of the cell. Some of it appears to be brought into the cell from its parent cell, so to speak. And where that information is stored, we don't know, right? How does the cell know how to build a new organelle like mitochondria or lysosome or whatever? Well, mitochondria divide, so they kind of split. But some of these other organelles are built. How does that happen, right? We have no clue how some of that happens. So there's some thought that there must be other sources of information in the cell that govern some of these subcellular structures and how they're formed, because they reliably make the same structure. So how does that work? How do they get to that place? It's not all in the DNA. What we understand, though, is that DNA isn't just loosely in your cell as this double helix. It's all compacted and wound up. And so I've got kind of some nucleosomes wrapped up there for you to consider, where the DNA is on the outside, kind of coiled around here. And then what you see in the middle in those colors are histone proteins in a little core. And so the DNA is wrapped around that core. And this compaction of DNA is dynamically regulated. Consider the fact that in every one of your cells, if you have 2 copies of the DNA, so it's a normal body cell, you've got about 6 feet worth of DNA in one cell. And some of us aren't even 6 feet tall, right? It's a huge amount of DNA, all compacted into a really extraordinarily small space. And my analogy I typically give my students is I say, look, let's take this room. What if we had hundreds of miles of rope piled in this room? Right? That would be kind of, by analogy, the concept we're talking about. Just take miles and miles and miles of rope piled in here. You think we'd be able to keep that straight, keep it from getting tangled? But amazingly, our cells do that. They keep it from getting tangled. And there's a growing concept, and I'll try to talk about this later in the week, of what we call the 3-dimensional genome, where now we're starting to learn that parts of chromosomes tend to reliably and repeatedly associate with each other in domains that allow for expression of genes that are needed for a particular function. So there's sections of our genome that somehow end up in the same physical space in the nucleus. Again, another process we don't understand. But they end up there reliably so that the cell can make the things that it makes. Now, from an engineering perspective, it makes perfect sense, right? If I need all these proteins and all their DNA is in this section and it's all in one place, that makes sense, right? So from an engineering design perspective, we would predict that to be the case, and it appears to be the case as we study the genome.
So you're saying they're more conservative. They're going to be throughout time. They're going to be there all the time.
That's right.
Throughout long ages of time.
That's right. And they're going to be there dynamically regulated. So as cell types, as cells differentiate from the parental cells to the daughter cells into different forms that become the different tissues, that the shape of the genome 3-dimensionally is going to change, so to speak. In other words, certain parts of the chromosomes are going to be turned off, other parts are going to be turned on. But somehow certain parts tend to always reliably associate with each other, and we're still trying to figure out why is it that those end up in the same place. And in some cases it appears that, well, that is a group of genes that all get expressed in the same time, the same place. And some of that is on the same chromosome, and some of that is sections of separate chromosomes. So it's not just within one chromosome, it's other chromosomes, their DNA is all coming to kind of the same place. It's a concept if you search for 3-dimensional chromatin, 3D structure of chromatin, something like that, you might see some of that kind of come up.
So the turning
Go
on
ahead.
and off may
Yes.
actually in part be due to different associations of one chromosome with another. In other words, maybe it's
Could be.
certain associations occur early in ontology, ontogeny, and other associations later. And then that's how you're turning it off.
Yeah.
It's not a physical switch. It's
That's right. Yeah. No, and it's not. Now it is a physical change in space. So like
interesting.
this DNA that's all wrapped tightly here is being regulated by modifications to the histone proteins that are in the middle. And maybe I'll bring this up later in the week, try to talk about this. But those modifications are either opening up or closing off access to that information. And so while it's not a on-off switch, it is an on-off switch in that there's actually a physical change in the way the DNA is compacted. It would be like if I had a rope in here, and I actually have my rope in the bag. I should have set this up. One of the things I have my students do to teach them about topology and the way DNA supercoils is I tie a rope to a doorknob, and I have two. I usually find really big, strong students to do this because it's very funny. I would make the rope double-stranded, and I have the two different students grab opposite ends of the rope and start pulling apart. And what happens is it bunches up as they get closer and closer to the doorknob. At some point, they have to stop because they can't pull it apart anymore. It's so wound up, they just can't pull it apart anymore. And then I grab the rope from them. I have everybody applaud for them. They sit down. And then I let some of the tension off the rope because they were putting a lot of tension on. I let the tension off. And what happens is what used to happen in your kitchen when you had a telephone with a phone cord, right? It starts wrapping up really, really quickly. And that supercoiling, that's something that occurs in our genome, and it's dynamically regulated. And it has to be maintained at a certain state for access to the genes that are needed. And so we'll talk about that more later, but to me it's a fascinating thing. We're just beginning to kind of understand that. So how are we doing on time? Okay, we're great. So many of you have seen this, so this won't be new to most of you, but just the idea that our DNA is double-stranded, so we have what we call a coding strand and a template strand. Now, genes can be on either one, so it's not that there's one chromosome has just a coding strand and a template strand. Genes can be encoded in either direction. But from that, we build the information in the messenger RNA. So notice where there's an A here, there's an A. Where there's a T, we make that a U in RNA. Where there's a G, there's a G, et cetera, so on down the line. And then where we take that information and to make the protein, a complex called the ribosome is going to read this information on the RNA and place the appropriate amino acid in place. And so as it reads along the messenger RNA sequence, it's placing the appropriate amino acid in place on the chain. So that's what's going to build our protein. And this is why when we talk about mutation, if you change something in this sequence, it's going to change the RNA, and that could potentially change which amino acid gets put in that place.
When it is said that there is no new genetic information being presented into a species or into a family, what does that mean in correlation to or in relation to this right here, what I'm saying right now?
Okay.
What do they mean by that?
So in humans, just for example, we're not, when we're born, we don't acquire new genetic information. So there's not a new sequence that's built in us. Now, we do inherit from our parents about 60 to 100 new mutations that they didn't have. So thanks, Mom and Dad, right? And so we're each multiply mutant, so to speak, as we go along. But the actual information itself is not dramatically being changed. Now, some things are getting swapped around here or there, but there's no new sequences being formed in us as a result of those processes. I would argue that things are breaking down to some extent over time. And so I think that today, biochemically, we do not function at the level that maybe we should have in the past, the level that Adam and Eve functioned at, or even that Noah and his family functioned at. I think systems have broken down since then. I mean, if you take a computer system and you start changing lines of code, Dr. Tackett, does it get better or worse if you just randomly start changing letters? Right?
It may fail on the first
one. It may fail, yeah, it may fail. Which speaks to the robustness of the system, right? The fact that our system can absorb as much change as we've absorbed over time speaks to how robust this system is, because it still works. And we know that there are cases where it doesn't work, right? We know that there are mutations that are lethal, that do not allow the organism to survive. So we know it can break down to a certain point. And my wife and I we had 3 miscarriages before we had Jed and Lucy, our 2 children. And what we learned about my wife and her genetics and the changes that are in her, I'm still trying to figure out how she's even alive, sometimes. But just realizing that those changes can be so subtle and can lead to, the loss of a child. It's a very interesting process. So, but the fact that it works so well, and that we're alive and can talk about it, to me speaks to the fact that it's a very robust system. So that builds proteins for us. Proteins can serve a lot of different roles: structural, transport, signaling, catalytic. many other roles. Some proteins are even multifunctional, right? Some people they've got proteins they consider to be the Swiss Army knife, of the body, because it can do multiple things. They call that protein moonlighting. When they got a protein they know does one thing, but they also think does something else, they'll call that protein moonlighting, which I thought that's kind of a funny way to describe it. But then there are some proteins are enzymes. So I say here most enzymes are proteins. I always have to hedge because if you want to argue it, RNAs, there are some RNAs that are catalytic that we call ribozymes. But generally speaking, when we say enzyme, we're talking about a protein, so something that's made of amino acids. And these are what really get the work done in our cells. These could be transport proteins. They could be enzymes that catalyze the breakage or joining of molecules together, or some combination thereof. This happens to be a topoisomerase, which is what I study, topoisomerase 2. Which I think is one of the best enzymes in the world, but that's, I'm a little biased. But then we've got massive complexes, right? I like the ribosome. This is an RNA and protein complex, and it just boggles the mind. It's made of about 80 different proteins plus 5 RNA chains. The RNA chains are massive, so we're talking about thousands of nucleotides. How does this complex get assembled so reliably in our cells? Because we need, thousands of them in each cell, and you've got hundreds, you've got, some people estimate 100 trillion cells. Well, let's just say you've got 10 trillion. It doesn't matter, right? It's a ridiculous number of cells that all are going to need these. So this machine has to be built very reliably. But even what it does is amazing, because this is what's going to grab onto the messenger RNA molecule, and then it's going to grab onto these tRNAs, which are RNA molecules carrying amino acids, and it's going to put the amino acid from the tRNA onto a chain that it's going to build to make your protein. What's interesting is the tRNA molecule is an adapter, so it translates from the nucleotide language into the amino acid language. So if I go back to a couple slides ago, it's this concept here. The idea is the tRNA adapter serves this role right here in the middle of saying wherever there's a UCA, we're going to put serine. All right, so it's what's actually physically making that connection. What's amazing about tRNAs, we have a bunch of different tRNAs, but each one has one specific amino acid that it will grab onto, and that's all it carries. So what do you have to do that? Well, you have to have an enzyme called an amino acyl tRNA synthetase, which can recognize the tRNA and put the appropriate amino acids. So that means for every amino acid, you've got to have an amino acyl tRNA synthetase that knows which tRNAs it can go on. Are y'all lost yet? Because I'm lost. But this is mind-boggling complexity. And the reason I bring this up is because it actually gets to an interesting point. James Watson did not like Francis Crick's adapter hypothesis. Francis Crick proposed that there had to be something between RNA and protein. There had to be some way to get across that bridge. So he said there's going to be an adapter, which we found out was the tRNA molecules. James Watson said, talking about the adapter hypothesis, he wrote, "I did not like the idea at all. More to the point, the adapter mechanism seemed too complicated to have ever evolved at the origin of life." So he acknowledged that this is if this is what is needed, this is a huge jump to get to a living system. In other words, it's so complicated, he could not see a way for it to have happened. And that's why some of these guys believed, I think, in directed panspermia. So they knew that life couldn't originate here, so they said, well, we'll send it off out there and then let it come here. So anyway, Crick was right. So the amino acyl tRNA synthetases were those adapters. I thought this is an interesting comparison to make, just to give you a sense of how many parts and components we're talking about are involved in some of these processes. So here we have humans, Homo sapiens, and here we have Mycoplasma genitalium, which is one of the smallest parasitic organisms we know of. It has to have a host. It cannot make everything it needs, so it's got to have a host to kind of feed it. But just to kind of compare, so our genome's 3.2 billion base pairs. This genome's 580,000 base pairs. So a difference in scale. So base pairs meaning how many letters. So if we think of it as a book, our book is 3.2 billion letters long. Their book is 580,000 letters. So it's a big difference in terms of how much information is there. if you look at some fundamental processes, what does it take to replicate and repair DNA? For us, it's around 160 proteins. It takes at least 30 proteins just for this replication process. Mycoplasma genitalium has about 30 proteins involved in that. For us, transcription DNA to RNA is about 40 proteins, Mycoplasma genitalium about 10. Translation, go from RNA to protein, it's over 100. Mycoplasma genitalium about 100. Energy metabolism takes over 70 to make ATP. Mycoplasma genitalium about 30. Why do I show you this? What I want you to see is that even when we pare everything down to as simple as possible, it still takes a lot of components. What we know about Mycoplasma genitalium is they've actually knocked out genes in that organism. In other words, they've shut off certain genes to try to figure out which ones are critical to survival. And what they came up with is that basically they can get down to about 382 genes that you have to make that thing work. That's a lot of parts, so to speak. If we're talking about a car, and we said the minimal car that we can drive is 382 parts, right? That's still a lot of parts that all have to be in place, all have to operate, all have to fulfill their function, their role. And if you take any one of those out, it's not going to survive.
And it's a parasite, so
Yeah, that's exactly right.
live on its own.
That's right.
It's like having a car
we're giving
in the
it-
midst of a car system.
That's right. We're giving it a lot of things. We're giving it the gas. We're giving it. That's exactly right. So I say that just to say, even when we pare it down, and Craig Venter and some of those folks are really interested in organisms like this because they see this as kind of biotechnology. What can we do with it? So they're asking some kind of biotech type questions with it. But even when you pare it down, you still got hundreds of pieces that have to come together to make this thing run. And that's a big deal because what this says is that even if you pare down the engineering of the system to its base level, we got to have a lot of pieces together to make this system work. So
If you consider that to be a simple cell or simple organism, and then you think about evolution,
Right.
started with this one cell,
right,
this is simple.
Right.
It can't live on its own.
that's right, that's right. Yeah, that's exactly the point. Is this organism cannot live on its own. It cannot survive on its own. And so the question is, what's the simplest organism that can live on its own, right? Then we start getting into genomes that are millions of base pairs long, right? E. coli is 5 million base pairs. There's probably some that are a little less than that. But that's where we have to go, is to that scale. I mean, an order of magnitude more DNA just for the organism to be able to survive freely in the environment. And even then, it may be scraping by, right?
So when the word simple is used, we're talking about a cell that's looking at in a ballpark of 380 genes under laboratory conditions.
Right. So and understand, we're making a comparison here. Simple is still is a relative term,
it's
right?
relative.
This is still pretty complicated, right? If it still takes 100 proteins for this thing to make proteins, that's pretty sophisticated.
What's the simplest cell we've been able to find not under laboratory conditions?
This is it. So this exists in nature, not as 382 genes, but it exists in nature. So normally it has about 480 genes, I think. And so it's right around 500 genes.
So it's
and that's the simplest version that we find in nature. But it's a parasite, meaning it's living on a host.
So the simplest form we've ever been able to find of something simple is whatever we have of our own intelligent designs have done.
Well, I mean, you can find bacteria that live freely that have genomes of maybe just a few million base pairs. So yeah, so it's possible, but it's rather large compared to this. So yeah, it's a good question. No, it's a good question. So, this is not a definitive, but what I wanted to give you is just a perspective on scale and on the complexity and all the various things going on in that system. So what about complex cellular pathways? So think about our metabolic map. So cellular function is very dependent upon a lot of different complex pathways. And these are highly regulated. They involve a lot of different feedback and control mechanisms. And this is where you really get into the engineering side of things, is how are all these things interrelated and working together? And also think about cellular signaling. So within the human body, our cells not only have to be able to communicate internally within themselves, so to speak, but also with cells around them in the same tissue or maybe distant, right? Somehow my head has to tell my feet what to do. There's a whole communication network that has to take place back and forth, and if that doesn't work, then there's a problem.
And we've got to know the outside too.
Yes.
Environment.
That's right. That's right. So you've got a lot of things. So as I showed you earlier, we've got lipids, carbohydrates, amino acids, nucleotides, and then down here, this is kind of our energy pathway down at the bottom, where we're ultimately making, you can barely see it, but there's ATP being made down there. So all of these things are connected and interrelated, and there's feedback back and forth between these pathways. There's components here that are going back and forth. The other thing it's depicting that's hard to see is kind of what processes are related to the mitochondria, which would be down in this range. So that's kind of mitochondrial processes versus outside the mitochondria. So this map shows you a lot of different things, and I know it's a blur right here. I actually have rolled up over here, and I forgot to bring my tape. I was going to tape it up here, a 6-foot version of this. Just to show you there's a lot of different things going on. So we'll break that out later. Yes, please, go ahead. No, this is what we're here for.
I'm a Bible text guy, so
that's exactly,
this is
hey,
awesome.
you don't have to apologize. That's exactly
But
what we're here for.
How is this communication happening? Is it or being received, being communicated? Are we looking at like vibrations and
Excellent question. Excellent
the
question.
liquid
Well,
of the cell? Or?
yeah, let me show you an example. So let me step back here. So an example we can relate to, right? A while ago you had lunch, so it's almost gone now, right? So our body has insulin and glucagon that are hormones that help our body know how well fed we are, so to speak. In other words, that's when you talk about someone having diabetes, that means that system has been disrupted, and their body does not know how to appropriately signal when blood sugar levels are up or down, right? So there's a disruption of that. So what we know is that insulin and glucagon are hormones produced in our pancreas, and they're distributed throughout the bloodstream, and they regulate blood sugar levels very dynamically. So when blood sugar levels go up, insulin production goes up, glucagon production goes down, and that signals to cells in the body that they need to take that glucose up and utilize it. In other words, hey, we got lots of glucose here, we need to clear it, so they'll start clearing it. And that requires a bunch of different steps, and I want to give you an example of that. So insulin is a physical entity. It's a molecule, all right? It's a protein, and it's going through the bloodstream. On the surface of your cells, you have a receptor called the insulin receptor. It's very creative. And the insulin receptor, guess what it does? It binds to insulin. The insulin receptor binds to the Internal Revenue Service. This is insulin receptor substrate, very creative. So the IRS is a protein that binds to the receptor. So this is outside the cell, out here, this is in, okay? So outside your cell, you've got this receptor holding out there, insulin binds it. That causes a change inside the cell to where now this insulin receptor substrate gets activated. All right, so it receives a physical signal and it turns on. And what that does is it turns on a series of enzymes that convert things over and creates a cascade of events that do several things in your cell. One, it tells your cell, keep living. Two, it tells your cell, hey, we got glucose coming, we need to start making the proteins needed to digest glucose. Three is it starts sending the machinery, so to speak, needed, this GLUT4 transporter to the cell surface so we can bring glucose into the cell. So we start telling the cell, hey, glucose is out there, so it sends the transporter to the cell surface. The transporter gets to the cell surface, finds the glucose, pulls it in, and just keeps pumping glucose in.
The short answer to your question is most of them are chemical.
Yes, that's what I was going to say. That's where I was getting to, exactly. So I think there are actually biomechanical signals going on that we don't understand.
Certainly electrical.
Yeah, absolutely. So there's magnetic and electrical signals that are taking place in our body that I think are poorly understood. But most signaling is a physical entity, meaning it's a protein or a smaller molecule that might be an amino acid or some related derivative, and it is produced by a cell, sent outside of the cell, and then it can go through the bloodstream or through the extracellular space, circulate through our body.
And the actual information is held through electricity that's being...
Well, there's not information per se. So what I want you to think of the molecule itself as being the information. So that whatever receptor it binds to, the cell knows what to do with it. In other words, the insulin receptor is not going to do what some other receptors do. So you have literally hundreds and maybe a few thousand different types of receptors available to your cells to produce. And so each receptor or group of receptors is going to have specific substrates that it looks for. And so it might be this one's insulin, this one's glucagon, this one's acetylcholine, this one is GABA, this one is, glutamine, whatever. And each of those different receptors is receiving a signal, and then when it receives that signal, it has a specific internal cascade of events that it's going to initiate, right? So that is, in essence, the molecule and the receptor, so the signaling molecule and the receptor are what actually convey the specific message to the cell. Does that make sense? Yeah, that's a really good question. That's a very fundamental concept. But it bears out the point, and that is that there's a biochemical basis to this whole system. And the specificity of the messages being sent in the different molecules and the receptors that can receive that is going to dictate how all that plays out.
It's a fourth analogy would be if you have the room designed in such a way when you walked in, you pressed every button, just something pressed.
That's right.
So that button turns on that monitor.
Yeah.
That button turns on that projector. That button turns on the heat.
That's right.
And so it's the same chemical signal, but the response is built into the system.
That's right.
It's built into the cell. It's built into the...
Which is really fascinating.
It's
incredible. This is an abbreviated insulin pathway. In other words, there's a lot more going on. But why is it that there's an insulin receptor substrate and a PI3 kinase and a PDK1 and an AKT and a glucose transport? Why is it that there are all those things, that they are all interrelated, they're all connected, they create a cascade? Because once the glucose is brought in, that's just the beginning. Right? Then we got to phosphorylate the glucose, then we got to convert to fructose, and then we got to phosphorylate again, and then we got to cut it in half, and then we got to go. All these different steps that are on that big pathway chart I was showing earlier, all those things got to happen. And that each of those steps requires different proteins.
And that's in every cell.
And that's in every cell. So, all right. Mitchell says we're over. So I was going to talk about topolysomerases, but I don't have time. So we'll get to that. We'll get to that later in the week. I know. I know. We'll have to save that for another lecture, but we'll get. No. You didn't at all. You didn't at all. So I say there's layers of organization, and I think that if we can approach living systems, understanding the biochemical basis, it helps us understand what goes on in the layers above that. And if we can approach the system from that perspective, I think it can really bring some clarity to some of the questions. So, thank you all for being here. All right.